Lactose is the main sugar in milk and other dairy products consumed in cultures domesticating milk-producing mammals. As a carbohydrate, it can be used as a vital energy source for the body. In humans and other mammal babies, lactose is consumed through the mother's milk at an early stage of life. In some human cultures, lactose is consumed throughout their entire life. However, the human digestive system cannot digest lactose in that form. It must be broken down into its two components, glucose and galactose, which can both be absorbed by the small intestine. The enzyme responsible for the breakdown of lactose is lactase, which is found in the small intestine. After the lactase enzyme breaks down the lactose molecule's glycolytic link through hydrolysis, the two sugars are absorbed through the lining of the intestine and pass into the bloodstream, where then they may be taken up and used for energy. Humans without the lactase enzyme active have their lactose digested by bacteria, which causes bloating, gas, diarrhea, and other digestive issues.
Before the domestication of milk-producing animals, humans did not drink milk after they were weaned from their mothers. As in other mammals, the ability to digest lactose in humans disappeared after babies stopped drinking their mother's milk. Over time, some societies domesticated animals that produce milk, such as cows, horses, and goats. These societies began to depend on these milk-producing animals for food, and they began to drink milk from their domesticated animals even after they were weaned from their mothers. Since people were using milk as a source of energy, the ability to digest lactase became a selective advantage in societies with domesticated milk. Any human that could digest the lactose in milk could get energy and sustenance by drinking milk. Humans without that ability were unable to get energy from lactose. In times when food was scarce, humans who could not get energy from milk and milk-based products were more likely to starve before having children, so they did not pass on their genes. As generations progressed, with the humans who could digest lactose producing more children and passing on their genetic code more often, the phenotypic frequency of humans with the ability to digest lactose increased. In the societies without domesticated milk animals, or in societies where these animals were not used as often to produce milk for humans to drink, there was no selective advantage for humans who continued to produce lactase after being weaned from their mothers. So in societies without domesticated milk animals, the phenotypic frequency of humans who could digest lactose throughout life was very low. The lactase gene also affects the overall health of women, and women who are lactose tolerant are on average much healthier, which contributes to the selective advantage of humans who are lactase persistent. The lactase persistent genotype has the strongest selective advantage of any known human gene, but only in cultures that drink milk.
The phenotypic frequencies increased in all cultures that drank large amounts of milk. As pastoral societies spread around the world, so did the mutation that allows humans to continue to digest lactose. The lactose tolerance gene is most prevalent in Northern Europe, North America, the British Isles, and Western Africa. Before the advent of lactase persistence, human babies lost the ability to digest lactose after they were weaned. This was caused by a regulatory switch during turning the gene that codes for lactase production off. When the switch has turned the gene on, the DNA can be read and pre-mRNA is produced. The mRNA is translated into an amino acid sequence that forms the lactase enzyme. This occurs because the switch is responsible for the formation of the transcription initiation complex at the promoter, the DNA site right before the sequence that codes for the protein. Once the transcription factors have been accumulated, RNA polymerase can attach to the DNA sequence and begin transcription. When the switch turns the DNA off, this transcription initiation complex does not form and RNA polymerase cannot transcribe the DNA, so no protein is produced. In individuals who can digest lactose, this switch does not turn the DNA off after the baby is weaned. There are many different mutations that can cause lactase persistence because the lactose-tolerant trait developed separately in the separate cultures domesticating animals. There are 5 distinct known mutations that cause the lactase gene on chromosome 2 to always be turned on. In most Europeans, lactase persistence is caused by a single point mutation, thousands of base pairs away from the lactase gene. In addition, the amount of lactase found in the body is related to the number of copies of the lactase gene present in the genome. The more copies of the DNA present, the more lactase can be produced at one time, and the more lactose tolerant the person is.
The allele for lactose tolerance is the dominant allele. Any children who inherit the allele for lactose tolerance will express the lactase persistent trait. This contributes to the prevalence of the lactase persistence trait, since the dominant trait is much more likely to be expressed in a population, increasing the phenotypic frequency. According to Mendelian genetics, a population of heterozygous individuals will produce children in the phenotypic ratio of 3:1, which means that the children of two heterozygotes are 3 times more likely to display the lactose persistent traits than the lactose intolerant trait when there is no selective force present. In cultures that consume a large quantity of milk, the phenotypic spread will become even more skewed toward the lactose-tolerant individuals because they are more likely to survive. In human inheritance, the child inherits one of each chromosome pair from their parent. The parent cells underwent meiosis to produce gametic cells of 23 chromosomes each. During sexual reproduction, one cell from each parent combines to make a somatic cell with 46 chromosomes. Each child will have 2 sets of genes that code for the production of lactase. If either of these codes contains a lactose-persistent mutation, then the child will continue to produce lactase after it is weaned from the mother.